learning about antibiotics, we need to learn about enzymes and how they work, because enzymes are a big part of antibiotics. So they can either make or break something. So here in this first example, we see a lock and key enzyme, meaning that the active site, which is that triangular area, is not changing shape. It is the same shape throughout. And this enzyme is making something, meaning a small orange piece, that substrate, is turning into a big orange piece, the product. So here in this next example, we see induced fit, meaning that that active site is changing. It is making itself bigger to accommodate that substrate, which is a big orange piece. And now it is breaking up into two smaller orange pieces. That would be the product, because this enzyme is breaking it down. It is a break enzyme. Let's talk about sulfonamides and how they work. So sulfonamides work by being a competitive inhibitor. What that means is they bind to the active site of an enzyme. So here in this example, you can see that that purple sulfonamide is stopping and closing off that active site, preventing this orange substrate coming into it. So how that works is that the sulfonamide blocks folic acid production, which is vital for bacteria replication. So, as a result, decreased folic acid levels will lead to bacteria cell death. Now let's look at beta-lactam antibiotics and how they work. So here in this image, we see this blue piece that is come into the enzyme, and as a result, the active site has now changed shape, preventing this orange substrate from coming in and the enzyme doing its job. So what this process is called is it is a non-competitive inhibitor. Non-competitive meaning that it is taking over the enzyme, preventing it from doing its job, but it's not directly coming into the active site. It's changing the enzyme in a different way. That's why it's called non-competitive. It's not fighting over the entrance of that active site like a competitive inhibitor. So let's get a bit more technical with why it does that. So these beta lactams, they bind specific enzymes called penicillin binding proteins. And what these penicillin binding proteins do is that they create peptoglycan subunits. And these peptoglycan subunits are a key component for cell wall creation inside of bacteria. So the bacteria needs a cell wall to protect itself. So if it cannot create the cell wall or repair it, that bacteria will die off. And that's how this antibiotic works. So now we're going to talk about two types of antibiotics, fluoroquinolones and tetracyclines, and they both work by preventing protein synthesis from occurring. Now they have two different mechanisms of action, meaning they are two different ways of how they stop it. So quick review of what is protein synthesis. There's DNA in its double helix, and it gets unwinded and then split into two DNA strands. Then transcription occurs where mRNA is created based off one of those DNA strands. That mRNA then leaves the nucleus and heads for the ribosome, also called the rRNA. Inside of the ribosome, translation happens where that mRNA codes for a certain type of amino acid, and then that amino acid later turns into a protein. So fluoroquinolones, how they work is they inhibit type 2 topoisomerase enzymes. And basically that prevents the DNA from unwinding and turning into two different strands. That is why here in this example, you are going to see this orange piece and it cannot break down into two pieces because that's the DNA and it cannot break into its two strands. So that way, and DNA replication cannot happen because DNA cannot be unzipped. Now then, tetracyclines work differently. Tetracyclines work by basically closing off and blocking the ribosome from receiving that mRNA. So if that mRNA cannot enter in the ribosome, it cannot replicate into more protein being created. So here in that photo, you can see that there is this green piece that cannot enter the gray piece, which is the enzyme. So that green piece, that mRNA cannot enter the rRNA or the ribosome to create protein synthesis.